Next, cytosine antagonists, a number of proteins that inhibit the biological activity of cytokines have been reported. These proteins act in one of two ways. Either they bind directly to a cytokine receptor but fail to activate the cell, or they bind directly to a cytokine inhibiting its activity. The best characterized inhibitor is the IL-1 receptor antagonist, IL-1RA, which binds to the IL-1 receptor but has no activity. Binding of IL-1RA to the IL-1 receptor blocks binding of both IL-1 alpha and IL-1 beta, thus accounting for its antagonistic properties. So, production of IL-1R has been thought by some to play a role in regulating the intensity of inflammatory response. It has been cloned and is currently being investigated as a potential treatment for chronic inflammatory disease. And cytokine inhibitors are found in the bloodstream and extracellular fluid. These soluble antagonists arise from enzymatic cleavage of the extracellular domain of cytokine receptors. Among the soluble cytokine receptors that have been detected are those for IL-2, 4, 6, 7 and IFN, gamma and alpha, TNF beta and LIF. Of this, the soluble IL-2 receptors, which is released in chronic T cell activation, is the best characterized. A segment containing the amino terminal 192 amino acids of the alpha subunit is released by proteolytic cleavage, forming a 45 kilodalton soluble IL-2 receptor. The shed receptor can bind IL-2 and prevent its interaction with the membrane-bound IL-2 receptor. The presence of SIL2R has been used as a clinical marker of chronic T cell activation and is observed in a number of diseases including autoimmunity, transplant rejection and AIDS. Some viruses also produce cytokine binding proteins or cytokine mimics. The evolution of such anti-cytokine strategies by microbial pathogens is good biological evidence of the importance of cytokines in organizing and promoting effective antimicrobial immune responses. Some viruses also produce cytokine binding proteins or cytokine mimics. The evolution of such anti-cytokine strategies by microbial pathogens is good biological evidence of the importance of cytokines in organizing and promoting effective antimicrobial immune responses. The pox viruses, for example, have been shown to encode a soluble TNF binding protein and a soluble IL-1 binding protein. Since both TNF and IL-1 exhibit a broad spectrum of activities in the inflammatory response, these soluble cytokine binding proteins may prohibit or diminish the inflammatory effects of the cytokines, thereby conferring upon the virus a selective advantage. Epstein-Barr virus produces an IL-10-like molecule, viral IL-10 or VIL-10, that binds to the IL-10 receptor, and like cellular IL-10 suppresses Th1 type cell-mediated response, see the next section, which are effective against many intracellular parasites such as viruses. Molecules produced by viruses that mimic cytokines allow the virus to manipulate the immune response in ways that aid the survival of the pathogen. This is an interesting and powerful modification some viruses have undergone in their continuing struggle to overcome the formidable barrier of host immunity. Then cytokine secretion by Th1 and Th2 subsets. The immune response to a particular pathogen must induce an appropriate set of effector functions that can eliminate the disease agent or its toxic products from the host. For example, the neutralization of soluble bacterial toxin requires antibodies, whereas the response to an intracellular virus or to a bacterial cell requires cell-mediated cytotoxicity or delayed type hypersensitivity. A large body of evidence implies differences in cytokine secretion patterns among T cell subsets and as determinants of the type of immune response made to a particular antigenic challenge. CD4 plus TH cells exert most of their helper functions through secreted cytokines, which either act on the cells that produce them in an autocrine fashion or modulate the responses of other cells through the paracrine pathways. Although CD8 plus TCTLs also secrete cytokines, their array of cytokines generally is more restricted than that of CD4 plus TH cells.